To how to put together a transfer sandwich. First things first, you need to put your nitrocellulose membrane and the transfer pads into transfer buffer at least 5 minutes before you actually take out of STS page. STS page I just pulled out of the beer cooler at that power source. This angle might be a little weird, but I want to show you how to assemble a transfer sandwich correctly. Things you will need for this process: obviously this stuff, your cassettes, empty clean tube, razor blade, or scalpel, depending on your method. The protocol, the most important thing is the order: black, pad, filter paper, gel, membrane, filter paper, pad. And then one of these, you want your same labels here. For me, I'm just going to do odd, so 3 is also 1. This is important for later in case anything goes wrong. Kind of deal. Boom. Okay, so I'm going to flip the angle and we'll see what happens. So these first, just gonna gently squeeze out because I don't have the other one that's clean, so I'm just gonna put over here. Also going to take these 2 sets out, just gently set right here. So wait for just 1 second. SDS, what I do is usually dump off a little of the liquid into the bucket of the water container first, just so it's less messy. Now you have this. You will pull out the middle portion. Sometimes they can be a little stuck. You will open up the 2 front gates to drain the excess liquid. Let me show you these 2. Make sure you keep it in the same orientation. So then you will pull up this guy to just your just. Now that you have this, I'm gonna set this aside. Dry off my hands. I like to do this on a towel because it's pretty messy. And put this here. This is full of our transfer solution, so you do black side down. Then I need one of the pads and the filter paper. Right there. And then I like to grab this. First thing I do, even before I put it in, just pop open the sides. Be just a little strategic on that. Now that you have it open, you want to submerge, and then you will gently peel off the top plate. Not too bad. What I like to do next is lift this back up, drain off some of the liquid, and you're gonna cut off on here all the wells. I just put them on the bench side and clean it up at the end. You can also cut off the sealant. Sometimes I do, sometimes I don't. Then I like to run my razor blade right down the side, usually twice. Now that you are in here, you're going to gently set it back down. Now that it's in the water, you should be able to slide it right off. I'm using 2 fingers to get the gel off. And you peel this off. Now, should be filter paper, gel. You want to make sure that it is aligned properly. So one hand's holding. Now I'm going to grab my, block membrane, set it on top. Your gel may move a bit. You need to make sure your gel is in place. Honestly, half the time I just kind of pull it out, gently move it around. You set this on top. Next is another piece of filter paper, and then the pad. Now that this is all together, you are going to submerge again. You need to use your tube and press out all of the bubbles. You want to do this, I usually do it twice. Careful not to move anything. And then you will push down and then pick it up. Kind of smash your fingers, it doesn't hurt, like this. And then close the transfer cassette. Loading it in here is very perfect. Black to black. So, let me, sometimes it takes a little maneuvering. Slide it in, like so. Then I'll put the other one black side facing forward, clear side facing red. You can fill it with transfer buffer. I usually wait till my other one's in there. I'm gonna do the second one. And then this will go to the power source in the beer cooler at 30 volts for 960 minutes, which is 16 hours, which is overnight. This is the hardest part for me, so good luck.